First, I think I have to make a confession. As you can tell from the title, I have managed to sneak in a talk about extant rats into our island symposium on paleontology, so sorry for that, but I hope that I can show you in the next 10 to 12 minutes that this sort of research is also very interesting for paleontology and that you can apply these methods for your paleontological research questions. So, I'm using 3D surface texture analysis as a method to quantitatively evaluate dental wear. If you're not familiar with the method, you can think of it as a sort of 3D microwear. But in contrast to microwear, it does not only take into account the pits and scratches on the surface, but the whole surface you're looking at, and it quantifies the profile, so the roughness of the surface, on a nanometer to micrometer scale. So far, it has mainly been applied to extant and extinct large mammals, but recently it has also been transferred to small mammals finally, and this is where I want to follow up with this study. So surface texture analysis is really great for reconstruction of diets, and if you want to compare different dietary adaptation between different species, and also to indirectly infer habitat parameters. But of course, you can also take another approach and use it for testing for intra-specific differences. And there's quite a number of studies back from 2D microwear and also in 3D surface texture analysis that have successfully found seasonal differences within one species or a high variability or dietary flexibility within one species. But today, I would like to show you what we did in contrast to other studies. Because we were not interested in seasonal differences, but in finding differences in dietary quality and composition between natural habitats and between habitats that have been anthropogenically altered, so changed habitats. And so our main research question here was, is anthropogenic influence on the habitat reflected by tooth wear? And you can find all the details in our paper from the March issue in Ecology and Evolution. And so we used a very generalistic rodent, the black rat, to test our idea. The black rat is pretty useful for this because it feeds on a variety of dietary items like nuts and seeds and other plant parts, but also on insects. So it's very flexible and can therefore easily adapt to new habitats and also invade new habitats, and that's why it's so widely spread all over the world. So the black rats have already reached Madagascar during antiquity, so that's at least what genetic studies show us. And nowadays, they're present all over the island, so in all habitats. We have captured rats from 4 different localities. 3 were located here in the western part of South Madagascar on the Mahafali Plateau. This is a relatively dry area with a mean annual precipitation of about 400 millimeters. And a 4th site where we captured rats was on the eastern south coast, a very humid area with a mean annual precipitation of 1,600 millimeters. So these 3 localities here are very close to each other, but very different in habitat structure. We have a completely natural habitat. This is the original vegetation in this dry area, a spiny forest. We have a manioc field, so an agricultural area, but there is still some vegetation present. And we have a typical village where really none of the original vegetation is left. And on the other hand, we have sampled in this rainforest here, on the east coast, which is natural rainforest, not really impacted by humans yet. So overall, we could use 37 black rat specimens for our analysis. Unfortunately, only 3 from the spiny forest and manioc fields, so I'm not going to put too much weight on these 2. But we had a very good sample from the village with 19 individuals and from the rainforest with 12. Animals were captured using Sherman life traps, both on ground level and 1.5 meters height. The sampling season was between April 2013 and November 2014. And our guys on Madagascar made sure that only Rattus rattus was captured. And And actually, they did not even trap any other rats, so maybe other rats are not even present on Malagasy yet. The maceration of the skulls took place in Hamburg, and there we also conducted the 3D surface texture analysis. And that's what I'm going to talk about now into a bit more detail. So surface texture analysis can be done using different machines. There is different techniques to do it. Interferometry is one. We're using confocal disk scanning, and this machine here, which is called the MeSurf Custom, made by Nanofocus. The pretty cool thing is that we have developed a very good routine in Hamburg, a filter routine to process these surfaces, and also a big database to compare our results to. So the usual procedure is that you take 4 non-overlapping scans on the enamel. The measurement field is 160 times 160 microns, and then a mean is calculated. But rats are a bit smaller, so as I have to show you now, we had to alter the method a bit. For rats, we are using the upper M1 and measure on the distal enamel band at the central position. But the problem is that the enamel band, as you can see here on an original scan, which is not filtered, the enamel band is actually smaller than our measurement field. So what we have to do is we have to cut small scans out of one of these scans. So this will be a filtered and leveled surface, and you can take small subscans to, well, try to represent the original surface best. And then we compute 30 standardized isonormed roughness parameters, and these roughness parameters are used to characterize the dental wear. So here I can show you what these surfaces look like afterwards in a 3D simulation. So I would now like to show you the results for the different habitats we sampled in. Not for all 30 roughness parameters, but for 8 of them. And I'm not going to go into great detail, but what you can see here, on the x-axis we always have first the man-made field, followed by the spiny forest, rainforest in green, and village sample in gray. And on the top we have height parameters, and on the lower column here we have volume parameters. But I don't want to discuss what they mean, because generally they mean all the same. If we have high parameter values, we have a high surface roughness. And so low parameter values correspond to low surface roughness, so relatively even surface. And as you can see, there is a general trend that is present in all these parameters. We always have a highly significant difference between the village sample and the rainforest sample. And I would like to zoom in into something to remind you that the village and these other 2 sites, the manure field and the spiny forest, were in the dry area. But what we can see is that here the median values actually for manure field and spiny forest are much closer to the rainforest than they are to the village sample. So what does that all mean now? If we organize our surfaces from high roughness to low roughness, we would have the highest roughness in the village sample, followed by the spiny forest and the manure field, and then the rainforest. High roughness in large animals is found in grazers. So high roughness corresponds to highly abrasive feet. And abrasiveness can be caused by different means. It could be external abrasives like grit and dust. It could be internal abrasives like phytoliths. But generally, a bulky and rough forage causes a high surface roughness, while a less abrasive forage causes a low surface roughness. And in these rats, we see a pretty cool thing. We see huge differences between the rainforest sample and the village sample. And these differences are comparable to the differences we see between grazers and browsers. So it is as if we were looking at 2 different species with completely different dietary specializations, which is pretty cool for a generalistic species, I guess. And what's also interesting, as I said, villages are in the arid and open area. The rainforest is more humid. Of course, we have a signal here to detect closed and humid and open and arid environments. But this is not only a pure aridity signal. Aridity itself is not sufficient to explain the high surface roughness in the villages. Otherwise, we would expect that the spiny forest and the manioc field, which are also in the arid zone, would be much more closer to the sample, but indeed they are closer to the rainforest sample. So what we get here is a signal between more natural diets and completely unnatural diets, because in the human environment they adapt probably on feeding on litter, leftovers, food from storage chambers. And we think that because they often feed on waste and litter from the ground, this is covered in dust and soil and sand, and so this high roughness, the high tooth wear, is actually caused by external abrasives consumed with the human waste. So in conclusion, human food is pretty bad for red teeth. Now I promised I want to say something about paleontological approaches. So we could show that 3D surface texture analysis is really good to use on small mammals, and it can also be used, of course, on fossil small mammals. For example, to assess niche partitioning in urban communities or habitat-specific, intra-specific differences on an island between two distant populations. So I brought you a little riddle down here. One of the surfaces is a Miocene giant dormouse, one is an extant rainforest rat. Would someone like to take a guess which one's which? Okay, so this is the Miocene sample, this is the rainforest rat, and I guess no one can tell the difference. So the preservation of these specimens can be so good, they are perfectly valuable for this kind of analysis. So that's all I want to say. Thank you very much, and I hope we have time for questions.